Okay, so what we're going to do today is we're going to go through a little bit of lecture type content. We're going to start walking through the tree of angiosperms. So we're going to start talking about the groups as we currently think they're related to each other based on the genetics, things we observe about the plants themselves. We'll be moving through from the groups that separated out first and moving towards our eudicots. So this is a different way of grouping plants than the way you'll be setting up your presentations, where you have specific adaptations that you're looking at, specific sort of plant lifestyles. Okay. So today we're going to talk about some of our sort of oldest living groups. I want to be careful when I say this, because when we talk about something being basal, what we're saying, if we're talking about a living organism, is that it's split off longer ago, not that, you know, it is necessarily a living fossil and nothing's happened in millions of years. So when we say that they're basal, that's the kind of context, but we often sort of simplify that and say that they're simple or the oldest type, or use kind of terminology like that. Just want to be careful that you know the sort of scientific context, because those are all kind of generalizations that we use to simplify a concept rather than the exact right way to do it. Okay. So our basal angiosperms are going to have 3 groups of them. We're going to move into 2 other somewhat basal groups, the Nymphaeales and the Nymphaeales. So looking at our tree, we had our phylogeny of land plants that we built in a previous lecture. So in green, we're looking at all of our angiosperms. So that's where we're cycling in. So when I say we're going to step through this tree, I mean we're going to go out like by branch by branch. So we're going to start with our basal angiosperms now. And then we're going to move through each node. So then the other 2 groups we'll talk about today are the Nymphaeales and the Amborella. Okay. So we are going to start out by reading a paper on the first flower and what we think the first ever flower looks like. So you can go to the link, or you can use my printout and draw copies of this article. And I know reading scientific articles can take a long time. I guess it depends how fast a reader you are. I remember at grad school, sometimes it would be a whole week to read one article, and I was actually trying. So you are welcome to read this whole article sitting here. You are faster at this than I am, but if not, I will out the most important sections, I think, that will help us get kind of a gist about what's going on. If you have a list of like vocab terms we've been going through, this would also be a great time to take it out, because as you read through about what this flower looked like, we're going to get very specific in describing it using our scientific terminology that we've learned about how to describe these plants. You're going to all read the first paragraph of the introduction to kind of set yourselves up, set the stage, and then split up the following sections. It's okay, depending on how many people are at your table, 2 people read the same section. But you're going to be looking for these questions at the bottom. You want to know what the authors are actually asking, what kind of data they use to answer their question. Telling you they're going to describe a flower, so I want you to tell me whether you still have this flower today. Tell me a little bit about parts of their results that they're less certain about. So when they use this word reconstruction, I don't know if you've defined that before, but when we say that we have a reconstruction of a fossil or something that was what we mean basically by reconstruction is like, it's a guess. It's a guess based on data about what this looked like. So they note some of these parts of their flower description, that they're a little less sure about and that are a little less statistically supported and some that are more supported. So we're going to take a look at those. And we're going to look for some stuff about the flower shapes and whether the flower is bisexual or unisexual. I didn't say that these are going to be their conclusions, but overall, basically, these are some of the things that they're concluding in this paper. If you have other questions or other things you think are interesting, for sure talk about those. And after you guys have this discussion, I want you to vote on which section of this you found the most interesting, because I want to know which parts you're most interested in. Okay, so I'm going to be quiet for a little bit while you split up who's going to read what, do some reading, and do some discussing.
I was gonna say my section really didn't have a lot. Well, I read the essential. Yeah, I was trying to answer to the question. Oh, there's some information. Yeah, they reconstructed it as a concept profile, but they never like figured out if it was supposed to be. Yeah, yeah. Oh. Yeah, because it's just about like how the world. I literally just talked about how they used, like the floral traits from Angiosperms as their data. That's literally all it's about. It's the, it's the office selection. And like, trying to one is the origin of Angiosperms and the third most important is Angiosperms. Yeah. Like Basal and Yeah. Oh, I'm watching. Oh, it's so massive. I think I see that as
Dominican Republic. Baseball. It's all. I'd rather watch all.
Okay, let's talk about this paper. Sarah, does anybody have any general comments? Right? Super in depth. Did anybody notice all the math and algorithm stuff I had you skip? You know, there was so much math that you needed to study to know about flowers. That was my takeaway. Anything about this surprising?
Yeah, because it's like, oh, we have it into direction of the future.
the impression, although I have no idea if this is true or not, right? But I'm always on the lookout for like science fights, you know what I mean? Like where there's probably like two different camps of people arguing about something. And I suppose I should say, what does it, everybody get the question? What is the question on this paper? What was it? Yeah, yeah, what did the ancestral flower look like, right? I suspect that if they put a paper in Nature about what did the ancestral flower look like, that must mean that they're disagreeing. But ever gets the details on it. I can tell you that human evolution people do absolutely fight about stuff like this. What was the result of your votes on interesting sections?
We said the first chunk that the ancestral flower.
Yeah, what's that? Anybody think anything different?
I'm talking about just the results section in general.
Overall, yeah. Yeah.
Most simple.
Yeah, I really like this picture. I think this is an amazing figure, right? It kind of summarizes the whole thing right there. Yeah. So, is this a living flower? What do we got? No, right? As you'll see, that part of why we're reading this kind of forms the basis for some of the groups we're going to talk about in like the next couple seconds. Yeah, looks like they expect an on A. Yeah. That's still like they don't know, but, so do you want to tell me what their data looks like? I had this question as I was starting this paper. But
That's it. You just look at all series as well as like.
yeah,
From living plants to the children. But then also like that and work from there.
yeah. Anybody pick up any more details on that? I get it. So I think they're looking at 21 traits, 790. So it's kind of an interesting thing. They talked about how they had a lot of missing data that anything that doesn't have a flower or doesn't have a flower for some of these traits, they have to like throw it out.
Yeah, they also talk about like if the traits only one, you'll be in your turn, but not all of them. They don't care that they're just figured out that that's the trait.
Yeah. Yeah. And they like tried to pair it up with molecular stuff, but this is stuff that they just went out and said it didn't go. Something I was like looking for in a paper, this that is for sure a science site, like whether you should be looking just at the genetics and the genes and stuff, or whether you should be looking when you're trying to reconstruct. You did about before. For uncertainty, anybody? What was uncertain here? Or like know where you looked it up? Yeah, I think that was one of the things that I'm certain about. And if you look at this, my favorite figure, anything with just one star, right? They marked how certain they were about these things, basically. Presumably based on their values, although I'm not sure they're very specific about that, but that's common in papers, right? So use little asterisks or key values. More in center. So you can see that they're pretty sure that the diamond skin is superior and have more than 5 sparkles, but they're not very sure that it has more than 10 tuples. What about the shape? First off, does anybody remember what these words mean? Axonomorphic and zygomorphic? Like what they're about generally. Yeah, it's about the symmetry. Yeah, so it's about radial symmetry versus bilateral symmetry. So like, can you put a line through it anyway and have it be symmetrical, like a square fit? That's the axonomorphic for radial symmetry. And the zygomorphic is like you have to draw a line very specifically in the middle. So which one do they think this flower was? And is this flower bisexual or unisexual?
We reconstructed it as bisexual, but it says it's unknown whether or not it's actually bisexual.
Yeah.
And they chose to reconstruct it as a bisexual.
Yeah.
Or what it is.
Seems like at different parts of the paper there might be like we talk a little sort of back and forth. And I almost wonder if that's a fight between that. How many authors there are? But I don't know if you noticed. They probably don't all agree. I have enough trouble getting three people to agree sometimes when I read papers.
Well, in their asterisks on next to the bisexual category, they settled on two.
Yeah. Yeah. Yeah, which is why I was wondering exactly what they think to the confidence. It's probably hard to qualify with the p value because I think they were looking at the. I didn't have you guys read through the methods they use. So this is kind of like the setup for what we're thinking about as we start to step through our tree of angiosperms. Okay. We're looking at how our living angiosperms diverged from something that maybe looked like this flower in this paper. So some of these traits that they identified here are traits that we're going to see in the groups we talked about today that give us a general idea that we're looking at kind of basal groups, which kind of supports this paper and kind of goes the opposite way as well, kind of supports the phylogeny that we picked for today. Remember, there are lots of different ways you can build a phylogenetic tree, and we picked the most recent one by an up and coming group, but there are lots of disagreements about exactly how plants are related to each other. Okay, so we're going to start, and I'll just go back here. So we're starting with these basal angiosperms. So that's the group that peels off first. So sometimes these are referred to as the anagrade, or in a lot of old literature, the ametagrade, which is just telling you the groups that are included in the basal angiosperms. So it's a couple of different orders, 3 of them in the phylogeny we picked for this class. The Amborella, the Nymphaeales, and the Austrobaileyales. So we got some pictures of them here. You can immediately see that some of them look like that flower we saw in the paper right over here. Some of them not so much. We're going to go through a little bit about what these groups look like, but these are the ones that are most different from our other groups of angiosperms, which I think is confirmed by both molecular and some of this observational morphological data. One species in particular that is regularly included in phylogenetic studies is Amborella trichopoda. That's because they use it as like the sister group for all of the angiosperms. So this is the one that's most on the outside of our group of angiosperms. But there's only one species in this group. We see it in islands in New Caledonia, which is in the South Pacific. It's a shrub, so we can see some pictures of it. So it's woody, not very tall, not a very thick trunk. It's got some pretty crazy to me looking flowers. It's got some groups of berries. It's got some toothed leaves here. So this is our earliest known living lineage. So it's the one that peeled off and diverged first. So it's useful when we're trying to look at the evolution of other groups of plants. Yeah. And some of the ways we know this is that a peculiarity is that the vasculature in the wood is different than most of the other angiosperms. So this comes back to something we briefly mentioned when we were talking about kind of gymnosperms versus angiosperms. The species doesn't have basal elements. So its wood is different and kind of more like other groups of plants than like all the rest of the angiosperms. A cool thing about this plant's genetics is that there's clearly been a lot of horizontal gene transfer with algae and lichen and mosses that are also growing in these shady environments. I saw some article that said for every like gene that actually belongs to this species, I think there are 6 that came in from somewhere else, from one of these other organisms, which I didn't get a chance to look into it, but it makes me a little bit wonder that it all influences the fact that it's peeling off as a sister group way early. So that's a good one to know because it'll show up on the outside of any of these sort of phylogenetic things. You can see what its flowers look like. So even though in that paper where they were reconstructing the ancestral flower, they did it as bisexual. One of the reasons there is some uncertainty, as Kelly was saying, is that this sister group that peels out and we think of as most basal has separated female and male flowers, so they're unisexual here. So those little ones are the female flowers and this big one is the male flower. Our next basal angiosperm is the Nymphaeales. So these are aquatic plants, 3 families of them, including the water lilies. So they're beautiful, they're cultivated. I'm sure you have all seen a water lily at some point, so you have all seen this order at some point. Thing to know about them is that even though they're floating on top of the water, they are connected to the ground. So they have root systems that attach to the bottom of the stream of pond they're growing in. But it's the rest of the structure of the flower that's floating. So they are tethered to the ground and push them too far. Their leaves can grow really crazy large. So a lot of them are small, but these are all pictures of the Amazonian water lily, which grows up to 2 meters, and you literally can sit on it like this. There's an actual park apparently in Taipei where you can go do it. So I have a new place on my vacation bucket list because I want to do that. So the reason it can do this is it has an incredibly strong vascular system, which they're looking at in some sort of bioengineering stuff. So here we see the underside of the water lily. This is not Photoshop, that's what it looks like. So you can see how it has this branching pattern. It gets thinner as it gets to the edge and not much space between. So they're looking into whether they can try using this type of construction to make stronger structures, maybe put solar panels that float on rivers. I saw someone suggesting. So it's kind of cool how we can look at structures in plants and in general in nature and come up with new ideas for what we could make based on them. So that's just one cool example of something in this order. Next, we have the Austrobaileyales. So there are about 100 species. So from our group of the 3 basal angiosperms, most of them fall in this group. In those 3 anagrade plants, there are only about 175 species total. So this is like more than half of them. So they're native to tropical areas in Australia, Southeast Asia, the Pacific. One of them you have probably seen before, at least in the grocery store, maybe. So star anise is an example of an Austrobaileyales. That's the Elysium verum, and it looks like this. So this is actually its dried seed pod, you can see. So you can see those multiple locules here, right? And those seeds inside. So star anise and other species in this group are often used for flavoring and perfumes and herbal medicine. And we're going to see that that's actually true of a lot of sort of earlier branches in our tree of angiosperms. One of the kind of commonalities, not true of everyone, so it's not a specific derived trait of this group alone, but it is generally true that we're going to find a lot of sort of essential oils and aromatic compounds in these more basal groups. In general, here we have some woody trees, we have some shrubs, we have lianas, so those are woody vines, basically. Their leaves are going to be simple, often going to be aromatic. They don't have stipules, which are those little sort of extra leaves at the base of the big main leaf. And their flowers are spirally arranged, which, did anybody catch that in the paper about the spiral versus whorled petals? Yeah. So part of why they were talking about that, right, is that in a lot of these basal groups, we see the floral organs, the petals, spirally arranged, but in their reconstruction, they came up with a whorled arrangement, which implies that this is a derived state, right? That this is something that was adapted, that evolved later, even though it's a commonality between these basal groups, between these ones that branched off earlier. So that's kind of a question mark about how or why that happened. We're going to step in a little bit now. We're going to talk about the Magnoliids and the Nymphaeales, which is really just one. So we'll talk about that one first. So we can still kind of group these things together as basal. When we're grouping things together, we have those anagrade ones, but the other two groups we focus on comparing are the monocots and the dicots, the eudicots, as we've spoken about before. So when people talk about basal angiosperms, sometimes they'll also talk about in the next sentence these two groups. All right. We have some common traits we might look at to figure out if we're looking at basically any of the plants that we're talking about in this lecture. That's why I've separated them out together. The anagrade plants, so those most basal angiosperms, or the Magnoliid plants. So if you're out in the woods, in a field, and you're trying to figure out what type of angiosperm you're looking at, we got a couple of things you can look at, right? You can look at the wood, you can look at the leaves, you can look at the flowers, you can look at the fruit, depending what's there. So if you're trying to do this, I'm trying to narrow down your group a little bit. You might note that these earlier basal, more sort of basic plants are going to have simple leaves, right? So they're not compound. We just have the one main leaf blade. They're entire, so their margin is smooth. Right? If we look at the flower, if we happen to have a flower, we'll see that it has many pistils, but they're unfused. So they're kind of clumped together here, but you would be able to separate out, sort of tracing along the edge of one at the base. So this is going to be different than a lot of our other more derived groups. You also notice that the stamen may be kind of flattened. So in our stereotypical sort of flower that we just draw like a little stick and a little pollen sacs for an anther, right? So our stamen, usually we draw as this sort of filament. So we do call that filamentous. But if we look at these more basal angiosperms, these are our stamens here. So they're kind of broader and flattened, right? They're not these little sticks, these little threads. So that's kind of distinct. And an idea about how this works is that these structures arose from leaves, right? So these almost look still a little more leaf-like than in our other groups. And we'd also be looking at the petals, the flower parts, but actually what we're looking at, as we discovered in our flower lab, right? So that sometimes what we think are petals are actually sepals. So we're just going to call them overall sepals, which just means we're talking about the petals and the sepals. So if you're in the field and it kind of looks like a petal, that's what we're talking about. So they're going to be spiraled around. We're spiraling, right, in a circle. Right. And they're not going to have a set number. So when we talked about monocots and dicots and before, we mentioned that one of the differences is that structures are in groups of 3 or maybe in groups of 5. If we're looking at these basal groups, there's no number we're going to focus in on, right? Like this isn't in any specific kind of grouping at all. Our plant may also be aromatic. So it may be smelly, it may produce essential oils, and it will often be pollinated by insects other than bees. So like those beetle eggs. So you wouldn't expect to see necessarily all of these traits or even most of these traits. This is not a hard and fast rule, right? There are lots of individuals that will be lacking one or more of these, and there will be some species within our other groups of angiosperms that do have some of these traits. But if you go through and you find more of these traits than you don't, that's a good sort of indicator that you should be kind of suspicious that you're looking at some type of basal plant, either an anagrade one or a magnolia. Questions about these traits? This may help you as you're doing your collections later on once it hopefully stops snowing. Help you narrow in on what you're looking for. So one family that we do kind of want to mention, this is not a terribly important family, but it is on our tree. So the Nymphaeales has a good fossil record on all continents, but right now, pretty rare. Some of the species in this family are used as fever reducers, so antipyretics are fever reducers, so that's kind of cool. Some of them you can drink as teas, probably you drink them as a tea to get the fever reducing properties. Yeah. But overall, they're tropical, they're soft-wooded, they're aromatic, right? So those are some of the traits already from that list. Their leaves are toothed, so that's a deviation here. They're evergreen and opposite leaves. Actually don't have petals at all. And sometimes they don't even have sepals. So you can look in on this flower, right? You don't really see a petal structure. You just see these sort of filaments coming off. There's fruits. They have either berries or drupes. Overall, not a super important group, but since it's on our tree, I wanted you to at least get a look at what it looks like. Bringing us to the magnolias, which are an important group to be aware of. So in much earlier phylogenies, this group was considered basically a group of dicots. That's why we've separated out eudicots now, because molecular studies and more recent research told us we needed to pull this out, and it actually wasn't that closely related to the rest of our dicots. So as we step through the tree, we will see that we'll go through monocots before we get to the eudicots, the true dicots again. Okay, so they're actually pretty distantly related from these plants that we thought that they were close to. So they have both woody and herbaceous species. So they have both plants where they keep their wood, keep growing year to year, and plants that they die back from year to year. We got 4 orders that we can zoom in on. We have the Nymphaeales, which are going to be giving this group its name, the magnolias, the Pennyales, the Laurelales, and the Piperales. So we'll see a couple examples of these things you might see out in the world and be able to brag that you now know what order they're in. Because that's obviously a brag. Okay, so they used to be considered dicots. I did see different arguments about exactly how many cotyledons you should expect to be here. Many sources say 2. Other sources say, it's not 2 that often, it's 3 to 4. So I'm always on the lookout for science fights. So I suspect maybe that's a science fight. TBD. Yeah. But they have some notably primitive features that contribute to that list we looked at earlier. Some other ones that we might look at is that their carpels are unfused, right? So that kind of goes along with those separate pistils. They either have many petals, right? So uncountable, not uncountable physically with your hand, but not a predictable multiple of anything, or they might have none. And the structure of their pollen grains and the pollen sacs is also kind of primitive. Most of them are found in tropical climates, but there are species that are found pretty much everywhere. So here we have some familiar, hopefully, examples. And if they're not familiar, you can wander around the grocery store or wander around outside again once it stops snowing and see some of them. So the magnolias that'll hopefully start popping up. Like this, a nice Easter looking flower to me. I know there's at least one in front of the that's the city hall building downtown. So I plan to be foraging some of its petals. I hear they taste like ginger. I've never eaten them. So I'm looking forward to when these start popping out. If you know where one is, we can talk. So that's part of the order Magnoliales. Cinnamon is part of the order Laurales. So the laurels, bay leaves, bay laurels are in this group as well. So cinnamon is actually the bark of a plant, the bark of a tree. So here we see that tree, and the cinnamon that gets peeled off in strips, where that comes from. So again, that has that sort of aromatic trait, right? And so does nutmeg, this next one. I had no idea that nutmeg was bright red. And we can see the fruit of the nutmeg here. So we actually have the dried seed that we use as a spice in the center. So that's also in the order Magnoliales. Then I would love to find one of these at the grocery store. I haven't seen one. I've never eaten one. I hear it tastes like pineapple sorbet. This is a cherimoya. Has anybody had one of these? Does anybody know what it tastes like? Well, if you see one at a fancy food store, let me know. I will go buy it for us and we can check whether that's true, because that was pretty exciting. But bunches of different types of fruits and common foods can be found here in this Magnoliales group. So some other examples here, we have another Laurales group. So we have the cinnamon in the Laurales. Avocado is actually in this same group as well. So here we see someone growing an avocado tree from their avocado pit. This is actually very easy to do if you have never tried it. Yeah, how big is it?
There's no. It's
Not yet. Okay. Okay.
There's two. The other one looks like it's dying.
Yeah, I had one that grew like a foot a month for like a little while, but it died during the pandemic because I left my apartment for 4 months. so someday I'll get another. But yeah, if you like avocados or you like growing random plants, I recommend trying it. It's pretty fun. You just put them in a little glass of water and they put down a root, put up a shoot. Black pepper is also in this group. So that's the order Piperales. So black pepper is actually sort of group of berries that is first green and then they are also red, and then the form that we eat is basically dried and ground up. I think it might even be a little fermented. I did look up the way you make black pepper, but I forget. So that's in this group. And a common houseplant, peperomias, are also in the Piperales. And what's 8? Well, not from one of my houseplants, but from a random plant that someone told me I could eat in the rainforest. A stalk that looked like this that I was told was in the Piperales, and my tongue burned for 2 days. I think I'm allergic to Piperales. Is part of why I'm very interested in exactly how black pepper is made. But yeah, if someone's peperomia grows off a shoot, I would be willing to bite it to see what it tastes like. Yeah, so those are some common individuals that we see in this group. Okay, so that's what we're doing for today. Any questions overall at this point?
Looked up that fruit and said it's only typically available in California.
All right, so I have to do another plant vacation.
We're going to
Yeah,
They have them in Spain.
they have them in Spain? Okay.